All right, folks, we're going to go ahead and get started. Good morning. Hope everyone's doing well. What a great crowd. What a great crowd. This is the largest Visions that I think we've ever had. There's over 500 people here at Visions, so thank you very much. I want to officially kick off and welcome everyone to Visions 2018. It's a very exciting time. Thank you to everyone who made the trip and is here in San Diego. Also want to thank and recognize the fact that for the first time, we're actually streaming this session and streaming many of the sessions from Visions live on Facebook and on the Eye on the Cure blog. So welcome to everyone who's participating remotely for this session as well. Our goal is to make accessibility number one in everything we do, and that includes bringing this great content to those that aren't able to make it here to the sessions live. So welcome to those who are watching remotely, and certainly a very generous thank you and a very sincere thank you to those who came and are here live in San Diego. My name is Jason Menzo. I am the new Chief Operating Officer here at the Foundation Fighting Blindness. Thank you. I've been here about 3 months, and this is my first Visions, and I'm really anxious to be part of this organization, part of this community, and looking forward to meeting many of you over the next couple of days. But first of all, I'm very excited to be part of this panel. So our first session this morning is called Mission Possible. This is a great starting session for the meeting because this is the session where we're going to be highlighting the top advancements in what's next in retinal disease, and there's no better audience to share that information than the four gentlemen sitting to my left. The presentations are going to take about 55 minutes. So what the format is that each of our presenters are going to get up and share a little bit of information. That'll take about 55 minutes. And then we will have 20 minutes for Q&A. And so as they're presenting, if there's anything that comes to mind, any questions that you've got, tuck them in the back of your mind, and we'll have an opportunity for questions and answers at the end. In addition to streaming the session live on Facebook and at our Eye on the Cure blog, we are also audio recording, so any of the sessions at all throughout the weekend that you are unable to participate in, you can revisit that content on our website in the next couple weeks. So I know that there are times in the agenda where there are multiple things happening at the same time, and you may want to be able to see what's happening in one session but not able to go. We will have all that content recorded and available after the fact as well. We also have assisted listening devices available, and in this room for this session, it's on channel A. So if you are using a listening device, it's on channel A. And just one other housekeeping note before we start, if you don't mind, please to silence your cell phones for this session. All right. Like I said, we've got four great speakers. We're going to bring them up here in just a moment, but what I wanted to do first was introduce each of them and give you a little bit of history about who they are and their expertise in this space. The first I'd like to introduce is actually Mr. Halden Connor. Mr. Connor is the CEO and president of Nacuity Pharmaceuticals. He's at the very end of the table to my left. He's also a member of the board of directors. Since 2013, Mr. Connor has led the development of NACA for RP as chairman of the board of Brighton Biotech, and their program was actually acquired by Nacuity. Previously, he's been an advisory board member of the Wilmer Eye Institute at Johns Hopkins, a board member for Alcon Labs. He was co-founder of Protom International, which is a maker of next-generation proton therapy equipment, director and principal of Private Access, which is a cutting-edge internet privacy company, and actually has a long legacy of nonprofit work as well. He was the chairman of the Hearing Health Institute, which is a nonprofit that performs infant hearing tests. He has his BA from Ohio University and an executive management program for Harvard. So thank you very much for being here. I appreciate it. Next, I'd like to introduce Dr. Daniel Chung. He is a former and senior investigator at the F.M. Kirby Center for Molecular Ophthalmology at the Shea Eye Institute at the University of Pennsylvania's Perelman School of Medicine. He spent more than 15 years advancing the science of viral and non-viral gene transfer and gene therapy for potential treatments of inherited retinal diseases. He trained as a pediatric ophthalmologist with additional training in inherited retinal diseases and gene therapy. Really importantly, he was instrumental in the RPE65 gene therapy clinical trials at the Children's Hospital of Philadelphia. He now serves as the clinical ophthalmic lead at Spark Therapeutics. So, Dr. Chung, thank you for being here. Next, I'd like to introduce my friend, Dr. Stephen Rose, who is the Chief Scientific Officer here at the Foundation Fighting Blindness, a man who really needs no introduction in this space. Dr. Rose is an expert in inherited retinal diseases. He is highly respected for his intelligence, his expertise, his humor, and his tireless commitment to finding treatments and cures for inherited retinal diseases. He has a PhD in microbiology. He's been with the Foundation since 2004 and oversees all of our research, clinical programs, our science department. He's just an amazing guy, tireless, and I'm really happy to be on the stage with him. Dr. Rose. And then finally, our first speaker is a new face to many. It's Dr. Ben Yerxa. Dr. Yerxa is the CEO, the new CEO here at the Foundation Fighting Blindness. I'm privileged to have known Ben we've known each other for about 10, 12 years. We've worked together in a couple different settings. And he's an amazing guy. He's a great leader. He's taken the helm of the Foundation Fighting Blindness and really has put his fingerprints on this organization in helping to drive us into the next generation in the future. He has a background in pharmaceutical development and biotech. He's a PhD. An amazing speaker, amazing leader, and I'm going to actually ask him to come to the stage and kick off this session. So, Dr. Yerxa, Ben.
Thank you, Jason. Good morning, everyone. I'm Ben Yerxa. It's a real pleasure to be here today as the, quote, new CEO of the Foundation Fighting Blindness. Not so new anymore. I say my honeymoon's over. I've been here since November, and it's a real pleasure to be here and to see this room so full of so many dedicated people. So thanks for coming out. This is my first Visions, so I'm curious to know if there are any first-time Visions attendees, if you wouldn't mind just raising your hand. Wow, that's amazing. Love it. Well, thank you for coming. It's great to be here in San Diego as well. This is my alma mater. I went to UCSD undergrad, so it's great to be back home in San Diego. So what I thought I'd do this morning is give you sort of an overview of the foundation, kind of where we've been and where we're going. So without further ado, starting with the beginning. So when we think about the foundation being founded in 1971, really by a couple of families, the Berman family and the Gunn families and others, got together and realized that no one was working on inherited retinal diseases at the scientific level. And it was really, that's a big problem when you realize that there's a big job to do here and no one's even working on the problem. So what they did is they got together and they funded Elliot Burson's lab, which is now the Berman Gunn Laboratory for the Study of Retinal Degenerations, and got this whole thing kicked off so that people would actually start working on understanding the basic science and the underlying disease causes so that we could attack this problem. So basically the foundation started funding the best minds in the industry and got them the funds they need to attack the problem. What's interesting here is when you think about this period of time, the National Eye Institute was only 3 years old. So even the NEI was just barely getting started. So this was, a long time ago, but great thoughts with our sort of founding families and donors to get all this work started. So understanding the cause of disease is really, really important. As you can imagine with retinitis pigmentosa, for example, where there are over 100,000 patients in the US, there are many genetic causes, and a couple decades ago, DNA sequencing, although you could do it sort of manually, it wasn't really automated and scaled up until about 10 to 15 years ago. So even though people were starting to work on genetic causes of disease, the rare orphan stuff really wasn't getting a lot of attention, and it wasn't until this was scalable to the way it is today that they could really fully understand everything going on in our space. And then you add in, Stargardt, Usher syndrome, Leber congenital amaurosis, many others. They're all unique and challenging, as you know, and it's a daunting task. When you roll it all up, about 200,000 patients in the US and more than 4 million globally. And if you add in the unmet needs in age-related macular degeneration, those numbers go to 10 million in the US alone. So it's a really big category if you think about it in its totality. But understanding the molecular cause of the diseases is the key starting point. It's ground zero. So testing new therapeutics is a big part of sort of what the foundation started doing. So The Clinical Research Institute was started some years ago under a different name initially, but really was set up to conduct clinical trials and support clinical work to understand what's going on here with a basic natural history of disease. And the foundation even funded a therapeutic trial of valproic acid, and although that didn't read out with efficacy, it was such a groundbreaking trial that the control arm became sort of a natural history study, and when all of our SAB members took a look at that, they realized there was a gem here. We found that there was an endpoint, an objective, non-invasive anatomical endpoint that could be used for clinical trials to study future therapeutics, and now there's been a whole bunch of publications on this. We've met with the FDA, and we've essentially validated a new endpoint that people can use to study retinitis pigmentosa. So that ended up being a really big accomplishment for the foundation. So measuring disease progression is extremely important. And so what we've done as a foundation is we have been funding natural history studies. A great example is one we just completed in Stargardt disease. It's called ProgStar. It's the largest natural history study ever done in Stargardt. Cost us millions of dollars, and it's about 460 eyes, studied over 2 years. And the foundation created the study team and funded them and the best minds in this space and said, go at it, come up with the best protocol possible, enroll the patients, and just figure it out. We just had a workshop a couple weeks ago, which was sort of the final readout of the data from this study. And I can tell you that, one, it was a very well-attended event from industry, FDA, academia, all sorts. And this natural history study has produced many viable endpoints for Stargardt disease for going after this with new therapeutics. So we're very excited about that kind of output, and we intend to do more of these as we go forward. So tracking patients in a registry, this is also an important part of what the foundation does. Many of you have heard of My Retina Tracker. This is really important to really understand the landscape of IRDs. So it's not an epidemiology study, projected numbers. It's real patients with real data. And it's important for everyone to know that it also comes with a free genetic testing study as a companion part of this registry. And that allows us to pinpoint the genetic cause in individuals. And of course, there's genetic counseling, and privacy, of course, is a very top of our mind, and we can pretty much guarantee the privacy here. But what you need to understand is that as the foundation gets information about new trials that come along, if you're in the registry, we can send out a note to everyone who might be interested, and just as information, whether you want to do it or not, at least you're aware of the options for participating in trials and just knowing what's going on. So it's really important to be enrolled in My Retina Tracker just for information alone. And for industry, it's also really important because industry needs to know that there are patients out there so they can do their trials. If you're a biotech company or you want to do some clinical trials in an orphan disease, if you don't have any idea about how many patients are out there or what their natural histories are like, it becomes a non-starter. So gathering this information in the registry is extremely important in sort of galvanizing industry to jump in and take on the task. So FFB is a great collaborator, and this is a really important part of what we're doing now and what we're doing in the future. So we're not only just funding and collaborating with our academic scientists, but we do things like we have a clinical consortium where we pull together 20 clinical sites all around the world, really the best clinical centers in this space, and we make sure that they're ready to go with all the right analysis and the right equipment, and it's all standardized and the protocols are all nailed down. And with that kind of firepower, we can jump into new natural history studies, which we just started one in Ushers 2A, for example. But it's also a way for industry, when they have new protocols that they want to get going, they can contact these centers and start enrolling and using them to enroll patients in their trials. Another important program for collaboration is the one that we have with the Harrington Discovery Institute. So we established something called the Gund Harrington Scholar Program, and that's a way for us to fund scientists and physicians in industry that have great ideas that need to be translated, and it's a way for us to create a future pipeline of projects that can get into the clinic in the next couple of years, and that's a very active program. We also work a lot with industry. We collaborate with industry on helping them with their protocols. We advise them on molecular mechanisms and just overall understanding of what's going on. And that's part of a service that we provide to industry because we have so much knowledge and we can leverage our scientific advisory board in order to make industry as successful as possible with treatments and cures. So in terms of convening, obviously convening our ecosystem, this is the premier example right here at Visions. Really, again, excited to see so many faces here from all parts of our ecosystem. But we also do this on a local level, so many of you have been to some of our Vision seminars that we do typically on Saturday mornings. That's also a great way to get involved and to get updated information, because we realize Visions are only every other year. The local Vision seminars are an important part of what we do to get the word out and to interact with our constituents. We also have a great conference now that's right before the largest ophthalmology research meeting in the world called ARVO, and our meeting is a Cell and Gene Therapy Innovation Summit. We had the largest attendance ever, even though it was in Hawaii, which is a long way to go. Okay, Hawaii, I know, you feel sorry for us. But it really was, it was a great turnout. I mean, the amount of companies and industry that were actively in our space in a really tough area of cell and gene therapy was just a testament to how active our field is right now. And it's become the go-to meeting pre-ARVO, and we're really excited about building that as kind of a flagship event in that research area. We also mentioned the ProgStar workshop already, but another important thing that we're doing now is we're meeting with other vision funders. So there are other organizations, besides FFB that are out there, and some of them have substantial resources. So we're now in active discussions with them about co-funding projects so that we can leverage our dollars and we can fund more projects if we can split them 50-50 with some of these other organizations that are funding some of the same labs. So that's a way for us, again, to leverage our hard-earned dollars to get to more treatments and cures as fast as possible. And I think it's fair to say the industry is fully engaged. They participate in, you see the sponsors around for Visions. We're really excited to see industry engaging, but also supporting the foundation and our events and all of our work here. So funding labs and projects. You're obviously aware that we fund the top researchers around the world, and we do this in a number of different ways. So we've been around for 47 years, so we've kind of figured this out. And the great thing about the foundation is they're not afraid to adapt the model with a changing environment. And that's something that we've done, I think, very successfully, where initially we had to just fund the top centers around the world to just figure it out. And over time, we realized now that some of these are moving along, there's so much action in the field, that we're moving to more of a milestone-based system. So we fund individual grants, we fund some program project awards that are longer and bigger, but they're all milestone-based so that they're shooting for certain deliverables so we can make sure that any money we invest into academia really delivers the results we're looking for so we can make as much progress as we can. There's obviously still a lot of work to be done. But our strategy is really paying off, because when you think about the pipeline, and if you've been watching this space over the last 10 years or more, you realize we're in a really interesting spot now where we're able to start companies if we need to. So we can, in order to get our mission done, if the best way to do that is to be aggressive with our funding and actually start a company, we'll do it. We'll do whatever it takes to get treatments and cures over the finish line. And a great example of this is a startup in Paris with Dr. Sahel's technology. We took a founding equity position. We have a couple of board seats, and we have, it's a way for us to be fully engaged and make sure that they're successful with that program. And so we're not afraid to take bold steps in order to get the job done. So here's the pipeline. When we started putting the pipeline together, and you'll hear more about this from Steve and others throughout the next couple of days, but there's so many new therapies going on, I couldn't get it onto one slide. So bear with me, and you don't need to read through all these. I'm not going to talk about all these companies, but what you need to know is that there's a great variety of people working in this field. These are the companies that are either in the clinic or poised to be in the clinic, within the next several months. And it's big companies, it's small companies, it's even academic institutions that are starting to run their own trials. So we count 30+ programs that are in the clinic or poised to be in the clinic right now, and there's a lot more coming behind it. So I think that deserves a round of applause. Here's page 2, and I think we have a handout for all these companies and projects that you guys can take a look at. But again, there's a lot of variety, and we're excited about the amount of engagement and frankly, the number of different disease targets that people are going after. It's a really active field right now. So in terms of other ways that we invest, we don't just do startup companies, but if we see a company that has a viable program and they're moving too slow or need some extra support, we will go in with a co-funding kind of environment where we'll say, look, we'll split the cost 50-50 if you can get the money to get something to a phase 2 result. And once you get a phase 2 result, it's a lot easier to get money from mainstream venture sources. So we've done this a couple times, both public and private companies. Nacuity is one of the companies where we're co-funding a project, and you'll hear next from Halden about that exciting program. And ProQR is a public company in the Netherlands that had a whole portfolio of a dozen or more programs they wanted to work on next. And we saw they had an USH2A program that was preclinical, and we said, hey, we'd love to work with you on that. Can you get that one going? And they said, well, if you co-fund that with us, we'll make that #1 priority. And so we did a deal with them in February, and that's now going to be in the clinic later this year. So we're really excited about how the foundation can leverage its dollars to radically accelerate the progress of these programs by working with industry and academia. So there's some great success stories. You're going to hear from Dan later on about this. But last December, we all witnessed the first approval of a gene therapy for an inherited disease in the United States. And that's really a remarkable achievement. And I still get goosebumps about this because it's so amazing. And what's really cool, this is the first. It was in the eye, it's in retinal, and it's in inherited retinal diseases. So what's happening now is the eye is sort of the proving ground for new technology. You hear about CRISPR and all these all this stuff. Everyone's proving the new technologies in the eye because that's really the best place to figure it out, because you can put the dose right where you need it to go. But here with Luxturna, we know it took a long time to get there for the first one, but now we've got a path. That path has been blazed, and hats off to Spark and the team there, because they had to be really creative. They had to invent new endpoints. The FDA had been stuck on, well, if you don't improve on a vision chart, we're not going to improve your drug. Well, they said, how about we create this new endpoint that's kind of a challenge test for low vision, and it worked wonderfully, and we're really excited about what they achieved. And like I said, it paves the way for new endpoints for future companies, but it also shows what's possible. They showed improvement in vision. And although that may not happen in every therapy we go after, that was a remarkable achievement. That really, in my world, I've never seen anything like it in my life. So really, really great result, and you'll hear more about that coming up soon. So finally, I'd just like to wrap up and thank everyone here. I want to thank all of our supporters and our scientists, for working on these hard problems for years and years, for all of you who've been supporting us either through donations or your involvement, walks, talking to people, spreading the word. It's really, really important. We really thank you for all of your support in these endeavors. All of our financial partners, industry, everyone. It's that whole ecosystem coming together that really makes it happen. So thank you for your attention, and I'll take questions later, and Halden, you're up next.
Good morning, and thank you, Ben, and thank all of you for allowing me a little bit of time to tell you about our fun little company. Nacuity was founded a couple of years ago only to pursue a small molecule called N-acetylcysteine amide, NACA for short, a very powerful antioxidant, and we're targeting the treatment of retinitis pigmentosa, although it may have some applications down the road. We're laser focused on RP. The joy of the company is we have a bunch of people that are exciting to work with and are some of the best in the country. It's rare that a small company has the opportunity to employ and enjoy the level of help we have. I am lucky enough to be from Fort Worth and have a long association with Alcon, which most of you have heard, is one of the top ophthalmological drug companies. And Jerry Cagle has been a friend for many years, and when I told him about NACA, he thought it was interesting, and so he's been very helpful. So all of a sudden, I'm the beneficiary of all of this talent from Alcon. Jerry and Michael Wall, who's head of our R&D program, and Fran Kane and Angela Cote and some of the best analytical people. It's really amazing that they allow me to hang around and watch what they do. I'm a good cheerleader. That's my primary, that and resources. Also, from a previous existence, I was fortunate to bring to bear Jim Herman and Doug, our CMC and preclinical toxicologists who have probably 10 years or more of experience with this particular molecule. So it's really been just sort of a magic coming together of exciting people. I almost cut this slide out, but my wife likes to be reminded periodically that I'm still employed, which is an important issue. Sandy Hillsberg is a 40-year friend of mine and has been very active in drug development and is an excellent chairman of our board. And of course, Michael Wall, I've already mentioned, runs our R&D. We are very excited to have these three gentlemen who are at the premier end of retinal research and clinical operations. Peter Campachero, I'll talk more about. David Birch in Dallas, who's quite active in the RP community, and Mark Panesi in Oregon. So now that you've seen all these fun people we get to work with, what excited all of them to come here in the first place was the science that was generated by this gentleman, Dr. Peter Campachero. Arguably, I think one of the top retinal researchers and clinicians in the world. And Peter, some 5 or 6 years ago, first published that it was his theory that, while genetic mutations were the underlying causes of retinitis pigmentosa, that he felt like it was primarily affecting the rods, and that as the rods died, they were creating oxidative stress, and was a situation that then started killing cones and central vision. So he put that to work in his naturally afflicted RP animal model and was quite pleased to find when we provided NACA that through ERGs he was able to get an indication that it was being very effective in saving the cones. Cysteine, of course, is the middle name of NACA, and cysteine is a key ingredient, as a non-scientist I'm told, that is required in the retina for glutathione production. And so he decided to test whether it was getting to the retina, and this slide indicates that in his animal models it indeed got right there. So that excited enough people and enough money, including FFB, to get on board to see if we could prove that this translated to humans. So this fancy chart, which is just a little piece of a great big PERT chart, as you can imagine, Dr. Wall, being a scientist, likes to try to schedule everything and know when it gets done. The magic little pointers only indicate that, excitingly enough to show that Dr. Wall's doing his job, we filed our IND, Investigational New Drug Application, last week. So that shows thank you very much on his behalf. So the team was very excited about that. As you probably know, that shows a massive expenditure of money and time on animal studies and toxicology and all sorts of things that are preparatory. So assuming, and I think it will be, that this is, the 30-day waiting period is over July 14th. So we already have our protocol ready to start a human trial in October with 100 RP patients around the country. Those primarily are going to be centered probably around the 3 advisors. They have lots of RP patients, and of course on my retina tracker we'll be hoping all of you are registered there. And we will be creating a cohort out of all of those, probably 3 locations, Baltimore, Dallas, and Portland, and possibly others, depending upon the need. As a small company, Ben requires that I watch the dollars fairly closely, so we can't afford to have 25 sites, but we'll do our best. The little markers that are on this chart are indicators of where we think we will get some indication of whether this is translating. Not enough for FDA approval, but enough to show those of us that have labored on this and invested in it that it's working in the right direction. So those little markers all occur mostly next year. So hopefully by the end of next year, 2019, we will have some indication, if I'm invited back, as to how are we doing. If they're not, I probably won't be invited back. So one of the significant things about this chart is that Ben mentioned alternate outcomes. I'm very excited that part of what's enabling us here is to have OCT as a legitimate outcome at the FDA rather than vision. Quite unusual, I understand, and I'll repeat something about that in a moment. So I can't end without telling you how important FFB is to this. Number one, we got a $7.5 million grant, which they've been religious to keep up with. We've matched it with private funds. In case you think that's all outflow, the nice thing about doing a grant with a private company like us is if it works, they get lots of money back so they can do this again, and that's important. But also, they're the ones that actually got the ellipsoid zone work done with the FDA to get us the OCT as an outcome, and that was a major accomplishment that they are to be applauded for. We meet with them regularly. They're a part of our thinking group. We were just up in Baltimore last week having what's called a Joint Development Committee meeting with all of their scientists and us. And all of their folks are very helpful to this outcome. It's really been fun to work with them, and it's fun to see if we can get something accomplished. Thank you very much.
Thank you, Halden. Really a tremendous advance. And I'm going to talk about now how the foundation continues to keep the pipeline going. You've heard from Halden in Acuity. We'll be hearing here in the room from Dan Chung from Spark. But I'm going to repeat a slide that Ben showed a little later in my talk. As Jason said, I joined the foundation in 2004, 13 plus years ago. I was 6 years old. But at that point, when I was addressing a group at Visions or elsewhere, it was always, there's great hope. There's a lot of good research ongoing. And we have to thank Gordon, Luli, the Berman family, and the other founders for taking on this challenge to understand retinal degenerations, to understand what is going on. For any of you who've seen the Illuminations video, Luli admits that they thought it would be a couple years and it'll be conquered. Well, those of us who've been in science for a long time know nothing is as easy as that, but they've stuck with it. All of the groups have stuck with it. The foundation has stuck with it. And as importantly, to be dead honest, all of you have stuck with it. Because without you all and without the donations that have been given to the foundation over the years, the research that is now leading to all of these treatments would not have been able to be done. So, as Ben said, we started funding the best minds, but the job is not done, and that's what I'm going to be talking about. While Dan will be talking to those of us in the auditorium about Luxturna, as Ben said, the first gene therapy for any inherited disease approved in the United States, there's plenty more work to be done, not only in gene therapy, but in cell therapy, genetic therapy. For those who follow the science, it's gene editing. And then Ben also talked about ProQR, which is antisense oligo work on RNA. And also, as we just heard tremendously from Halden, from work that the Foundation helped support with Peter Campachero for NACA, NAC, and the antioxidant. We're focusing on many different applications that will treat multiple inherited retinal degenerations, not just gene-specific. This is a slide many of you have always seen. We are not just retinitis pigmentosa. As Ben said, Leber's congenital amaurosis, and in this case for LCA and RP, the Luxturna gene therapy will work. But Stargardt disease, a very major emphasis in the Foundation Fighting Blindness, as is Usher syndrome, all forms of Usher syndrome, choroideremia, X-linked retinoschisis, and actually many others, including Bardet-Biedl, and some that you may or may not know. This slide just talks about Luxturna, but the point I'm making here is that it's not the only type of gene therapy. Luxturna is gene replacement. It is replacing the RPE65 gene that is not working in the cells. But there are other types. A drug factory, as Ben mentioned, sparing vision, RDCVF. The gene therapy there is to provide a molecule that's a neuroprotective. And then there's optogenetics, and that is to try and provide sight back to individuals who have no vision left by basically replacing the light-sensing molecules in the retina that are no longer there because the photoreceptors are no longer there. So this is a recapitulation of Ben's slide. This is probably one of the most amazing slides that Ben or I are going to show. And that is because there are so many clinical trials ongoing now, over 30 clinical trials in the inherited retinal degeneration space. No longer only talking about hope, but actually talking about trials that have the opportunity to provide a real treatment. And from this slide, the thing that's so important is it's not just in RP. It's in achromatopsia. It's in optogenetics, 2 ongoing clinical trials in that area, and there'll be a session on optogenetics with John Flannery and Jackie Duncan, who'll be talking about that going forward. X-linked retinitis pigmentosa, an area that we have long been involved in. Also, many different forms of RP, as well as Stargardt, the ongoing StarGen clinical trial from Sanofi, as well as Ushstat, the ongoing clinical trial in Usher 1B. It's just amazing, choroideremia, to watch how the field has really burgeoned going forward. But, as the title of my talk said, we're not done. There are many, many more ongoing pipeline of gene therapies. Dominant retinitis pigmentosa, a major type of retinitis pigmentosa due to mutations in the rhodopsin gene. We have a long-standing project on CRB1 RP. We've been funding for a long time in the Netherlands with Jan Winholtz. Obviously, Usher syndrome, tremendous advances made, ProQR with the Usher 2A antisense oligo, but there are other forms of Usher, Usher 1B, Usher 3, that are all under investigation, as well as Usher 1F, Usher 1D, and the alphabet goes on, unfortunately. LCA1, we have the privilege of having Shannon Boy here with us to talk about gene therapy. Shannon's work at University of Florida on LCA1, GUCY2D gene, has now been taken up by Sanofi for development. Another form of LCA, a childhood form of blindness. Tremendous advance with a company taking it forward. Stargardt disease, obviously I talked about StarGen, but there are others who are working on many different types of gene therapy or genetic therapy for Stargardt's, and then a plethora of other genes that are at the bottom as well, different forms of LCA, different forms of RP. This is gene editing, and I put this slide in, the only graphic that's there is just basically to show that it's a cut and paste. The idea in gene editing is not to put back a gene for one that's missing, but to repair the gene right there in your own cells, in the genome. Take out the part of the sequence that is not correct and make it correct again. That really, when you come down to it, as well as gene therapy works, and it does, as well as other therapies like NACA and others that we're going to hear about during the rest of the Visions conference work, obviously the ability to repair the gene right where it is so that it is regulated as it normally would as if there wasn't any change in the sequence is really would be a tremendous advance. Editas, CRISPR Therapeutics, Casibia, quite a number of companies are working. And as Ben said, the eye is leading the way in a lot of these companies. They see it as a great area to test because the ability to assess the success of the treatment is as easy as taking a photograph, looking at an eye chart, even though that's not an endpoint, but doing the OCT, looking at the structure of the retina, really makes it a difference. We have the saying, and you've heard it many times if you've been in our seminars, the eye is the window to the brain. The eye is neurons, which is the same as the brain, and it really makes a big difference to be able to non-invasively understand what effect the treatment is having. Making a mutant gene normal, that's what gene editing is about. There are quite a number of programs ongoing in gene editing in the eye, as I said, including Stargardt, LCA10, and others. Stem cells. If you've listened to us for many years, you've heard a lot about stem cells. There are two types of treatments in stem cells. One is to protect what you have. That's an ongoing trial right up the road. Almost said down the road. I'm used to being in San Francisco. At University of California, Irvine with Henry Clausen and J-Site. We funded Henry's work many, many years ago as he started this endeavor. This is now going into a phase 3 clinical trial and is showing some great promise. Yet to be determined how well it works on the overall, but it's looking very good, which is why it's going into phase 3. And then there's build what you don't have, i.e. replace the photoreceptors. As much as gene editing is definitely the way that we would like to go very early in a retinal degeneration, if you don't have any photoreceptors left and you're far along, stem cells, rebuild the retina, the holy grail. There's a clinical trial ongoing at Massachusetts Eye and Ear with Renuron, a company we had supported the early work with Mike Young at Scapins many years ago. All yet to be determined, but it just shows that the foundation is taking a broader view, a strategic view, not just a single gene replacement, but a strategic view in pan-therapies and providing potential for people to be able to see again in many different ways. There's a pipeline of cell therapies that are ongoing at the moment. We've contributed and continue to evolve the paradigm over its 47-year history. We initially funded centers of excellence, and the strategy is now paying off. That's what's really important. Why do I show a slide of tulips? I show a slide of tulips because the results that we are seeing now are resulting from the bulbs that the foundation planted many years ago. They are now showing the flowers, the treatments, the trials that are coming about. And it's because of the foundation support, due to your support of the foundation, that we are seeing all of these clinical trials, all of these successes, and in fact, what those of us in the audience here are going to hear from Dan Chung from Spark Therapeutics. Unfortunately, due to regulatory controls, the streaming of Dr. Chung's talk is not going to be able to be done. Spark is a publicly traded company, so the streaming will be turned off for a bit while Dan provides his talk here to those in the audience, but it will be turned back on for the Q&A session. And with that, I thank you all very much.